at the Mission-Aransas National Estuarine Research Reserve, and we're based at the University of Texas Marine Science Institute in Port Aransas. We have a project that's specifically looking at freshwater inflow in the Mission-Aransas estuary and watershed. It's funded by the NERRS Science Collaborative, and they fund projects that fund research to address related policy needs. So in Texas, freshwater inflow is one of the biggest issues that we're facing as citizens and policymakers today. And it's important because the amount of fresh water coming to our estuaries is vital to securing the needs of our fishes and crabs and other species, as well as habitats that we as humans depend upon. Texas has implemented a process called the Senate Bill 3 process that helps identify the amount of freshwater inflow needed to support a healthy bay and estuary system. The research that we're doing here at the Mission-Aransas Reserve is looking at what those freshwater inflow needs are and providing information to our policy and decision makers to make more informed decisions.
So one of the goals of the Freshwater Inflows project is to better understand how differences in freshwater inputs are going to affect salinities in different parts of the bays. And right now we don't really have any actual measurements of currents in the bays. So we're putting out these current meters so that we can better understand circulation within and exchange between the bays and the estuary. Right now we have the meters out at 15 stations within the bays. To determine the locations of these sites, we asked the stakeholders at one of the stakeholder meetings where they thought the meters would best be placed to capture the main currents in the system. We also had a meeting with local fisher guides to ask them where they thought the meters should go because they're sort of familiar with the currents in the system from being out on the water all the time. Between the feedback from the fisher guides and the stakeholders, we determined the 15 sites that meters are at now. So the current meters are these white rods that look a little bit like white PVC. There's a tether on one end that attaches to a PVC stake, and the meter is buoyant, so as the current pushes it, it'll lean, and it'll record how much it's leaning in the flow. At the top of the meter, all the data is recorded in this data logger. So we set it to log at a certain interval, put the cap on, and let the meter log out in the field for a period of 2 weeks up to about a month. So when we're trying to visualize these large volumes of data that we get from these current meters, it can be very difficult to look at all of it at once. So one of the best ways, I think, to visualize the data is to make an animation on a map where you have arrows representing the current at each station, and these arrows change in length and direction over time to match the current speeds and directions, and then you can relate the currents at the different stations to changes in wind speed and the tide levels, and you can sort of visualize the relationships that are going on. So the data from these meters will be used to calibrate this TextBlend model, which is a model developed by the Texas Water Development Board. The TextBlend model simulates currents in the system, so we can use this current meter data to calibrate the model and make it a little more accurate.
Blue crabs were selected as the focal species for this project for a number of reasons. First of all, the BBASC Adaptive Management Plan listed blue crab responses to freshwater inflow and blue crab life history and habitat studies as one of their top tier research priorities for the area. And then we also, at the first stakeholder meeting for this Science Collaborative project, participants ranked various focal species in terms of how important they thought they were for the estuary and how important it was to understand how they responded to freshwater inflows. And blue crabs were ranked highly by that group, so the combination of that high ranking and the BBASC Adaptive Management Plan further supported this choice. And then finally, they were partially selected because of their economic and ecological importance in the area. They support a large commercial fishery in Texas as well as throughout their range, and they're also an important prey species for various fish as well as birds, including endangered whooping cranes. We're studying several aspects of blue crab responses to freshwater inflow and how population dynamics respond to changes in freshwater inflow. So one large component is looking at blue crab recruitment into the estuary at the megalopal stage. This is the stage when they move from that offshore planktonic lifestyle into the estuary to settle into submerged nursery habitats like seagrass beds in the lower estuary. So we're looking at that influx of megalopy into the estuary using passive megalopal collectors that are placed in several areas throughout the estuary. So these collectors are sampled daily by volunteers. We're also kind of in another line of research looking at how juvenile blue crabs, so several weeks after that megalopal stage, how they respond to changes in salinity and different salinity regimes. The data from the megalopal collector study looking at megalopal recruitment is going to be combined with lots of environmental data from various weather stations in the area, as well as the current meters that are being deployed for part of this project, to look at what environmental conditions control the megalopy moving into the estuaries. And then the growth experiments, those are going to be primarily used to determine, to make predictions of if we see changes in temperature and freshwater inflow, which lead to changes in salinity, how is that going to affect blue crab growth? So our approach is really just to better understand what may happen in the future if freshwater inflow is altered, and what levels of freshwater inflow are going to be important to maintain stable blue crab populations.
We're using Ragia clams as an indicator species, and what that means is that Ragia clams tend to live in brackish waters. However, they need fresh water in order to survive and reproduce. So if we aren't getting enough freshwater inflows into our river systems, these clams might not survive, so we're using them as an indicator for that. What we're doing is we're going through and looking at the brackish water inside of the Mission-Aransas Reserve, and we're also exploring the surrounding areas where freshwater comes in, so the Aransas River and the Mission River, and just essentially trying to locate where these clams are distributed. No one's really done any studies in our area specifically looking for the Ragia clams, so we're obviously going out and trying to locate the species and figure out specifically what areas are a healthy place for those clams to maintain their population.
Our project is really unique because it's involving research and policy together as one. And that's important because oftentimes research projects will do research on their own and not really involve policy until at the very end. We're involving policymakers and people that are making decisions throughout the process. So in doing so, they're helping understand the research and driving what research is needed, and they're able to understand it and then use it into policy making decisions.